maker.
Yes. Here is our bacteria. We will find it's already become clotted. So that means our bacteria grows very well.
It's day 3. Haoyu is going to aliquot inoculated broth into 15 milliliter tubes. And in this case, Haoyu will use 16 of those tubes. And in each tube, there will be 5 milliliter of inoculated broth. The exact number of tubes and amount depend on your needs and your protocol. The process continues until you have 16 of those tubes.
So we will go to centrifuge.
We will spin it down to get the cells. And when you place the tubes, make sure that they arrange in a symmetrical way so that the force gets balanced. We use 40 to 100 RPM for 5 minutes. If your rotor has a higher limitation on the maximum speed that you can have, then you can go higher RPM and use less time. So it's flexible as long as you get the cell down.
It's a P1 buffer.
P1 buffer, You made it or it's ordered from?
It's ordered from our kit,
Okay.
but it needs to be stored in for.
Well, we are waiting for the 5 minutes centrifuging process to be completed, and we can do some preparation at the same time. So what Haoyu is going to do is take out the buffer we are going to use and also arrange the microcentrifuge tubes. In our protocol, what we are doing is we are going to put the cells in every two 15 milliliter tubes into one microcentrifuge tube. Since we have 16 15 milliliter tubes, and then we will need 8 microcentrifuge tubes. You may use a totally different protocol that require a different number of tubes and the amount you put in each microcentrifuge tube. That's totally fine, and you just follow your needs and do some modifications.
You could find it look like this. Here is a bacteria. Here is a media.
To isolate the plasmid DNA we want, we use a method called alkaline lysis to separate the small circular plasmid DNA from the larger tangled genomic DNA and other cell debris. The first step is to use 250 microliter P1 buffer to resuspend pelleted bacterial cells from every 215 milliliter tubes and then transfer them to a microcentrifuge tube. This P1 buffer contains RNase, an enzyme that degrades all the RNA in the cell. Without this, the RNA would be purified along with the desired DNA, which is not good. So we need the cell RNA to be degraded. The P1 buffer has lyse blue reagent. The next step is to use the P2 buffer to perform lysis. In each microcentrifuge tube, add 250 microliter P2 buffer to the resuspended cells from the P1 buffer. Mix thoroughly by gently inverting the microcentrifuge tube 4 to 6 times. Since we have added lyse blue reagent to the P1 buffer, the solution will turn blue. Do not allow the lysis reaction to proceed more than 5 minutes. This P2 buffer contains a detergent and a strong base. The detergent breaks open the cell membranes, and the base denaturates all the DNA from both plasmid and genomic into single strands. The solution becomes very gluey. Following the P2 buffer, we will add N3 buffer for neutralization. Specifically, we add 350 microliter N3 buffer to each of the microcentrifuge tube and mix thoroughly by inverting the microcentrifuge tubes 4 to 6 times. If using light blue reagent, the solution will turn colorless. This acidic buffer instantaneously neutralizes the base from buffer T2. This is the magic step. The small circular plasmid DNA quickly finds its complementary strands and snaps back together. The massive tangled genomic DNA cannot refold properly and crashes out of solution in a white, insoluble mass, along with proteins and cell debris.
Change to this one.
Now we will spin down all the white debris, such as genomic DNA and proteins. The plasmid DNA will remain dissolved in a clear liquid called supernatant.
It's nearly 10 minutes. I usually will prepare for the next step. PV buffer, and last one EB buffer. Like this.
For each microcentrifuge tube, remove the supernatant and transfer it to the spin column by pipetting. In the high salt conditions created by the buffers, the plasmid DNA sticks tightly to the silicon membrane while impurities pass through. centrifuge the spin columns and discard the flow through. Wash the spin column by adding 500 microliter PB buffer, then centrifuge and discard the flow through.
Setting. PE buffer. First.
Next, we wash the spin column by adding 750 microliter PE buffer, then centrifuge and discard the flow through. The PB and PE buffers wash away any remaining salts and impurities. PE buffer contains ethanol, which cleans the sample without dissolving the DNA from the column. centrifuge again to remove residual wash buffer. After placing the spin column in a microcentrifuge tube, we will elute DNA by adding 15 microliter EB buffer to the center of the spin column. The EB buffer is a low salt buffer. In this low salt environment, the DNA no longer sticks to the silica and dissolves into the buffer. You can let this process stand for 1 to 10 minutes depending on your need. We will wait for 10 minutes. While we are waiting, we can do some cleanups.
Just turn back. I need to use timer to calculate
After 10 minutes, centrifuge the microcentrifuge tubes to collect the liquid, which gives the final purified plasmid DNA. Now we have got the purified plasmid DNA we want. If you have the equipment and you would like to, you can also measure the density of the purified plasmid DNA.
Here is 2060 and 280 number. This lab. And this is the concentration.
Temporarily transfer the purified plasmid DNA to -20 and then finally to -80 for long-term storage.